Hi, my name is Sitarama Joyce, the project lead for the NIGMS Sandbox module from the Louisiana Institution Development Award. And Dr. Gus Kosalos is the PI for the IDEA Award, the LBRN. And here is my team who helped me to develop this module. The module name is Structural Biology and Drug Discovery. I'm going to give a brief introduction about this module. This is a cloud-based learning module for learning protein structure, structure prediction, structure-based drug discovery, and protein-protein interaction. Undergraduate students, graduate students, researchers who want to learn the structure-based drug design can take this module. And this module is developed in such a way that you can use it at your own pace. So it's self-learning. And the new users can start the module from submodule 1 and then go to 2 and 3. If you are familiar with the protein structure, the details of protein structure, you can go to module 2. Why need to study protein structure for the drug design? If you look at the number of drugs available in the market and where the target, the pharmacological action, you will see that 97% of the drug targets are proteins. And among these, G protein coupled receptors make the major part, 33%. You have nuclear receptors, enzymes, different kinases make 30% also, and ion transport proteins 18%. That's why looking at the structure of proteins is very, very important. What I'm going to do here is to show the GitHub repository. When you open this module page, this is how it looks like with different submodules arranged in a particular way and README file. And you have the contents, which basically background, overview, and how the module is designed, module 1 and 2, 3. And before you start this module, you need to know how to use the virtual machine. Once you do that, you can go to the module and start. These are the details of the module. Here is the submodule 1. You go through the basics of protein structure, some of the details, look at the protein ligand interaction as well as protein modeling, and you go through quizzes and assignments. In submodule 2, you will look at the basics of docking and docking of small molecules of protein, protein-protein interaction, and protein docking using online software. You go through the quiz and assignments. And in module 3, you combine the knowledge of module 1 and 2 and apply that to drug design. We cover enzyme-based drug design, receptor-based drug design, protein-protein interaction drug design with special preference to peptide-based drug design. And module 4 is optional. There are some additional assignments, challenging problems. If you like that, you can go to module 4 and solve some of those problems. As I said before, each one has a README file, step-by-step instruction, each module. And we have activities, quizzes. What are the tools used in this module? PyMol is used for visualization of biomolecules. And we can also use sketching and converting. This is specifically for small molecules. Sketch a small molecule, convert them to PDB file. Peptide structures can be generated using Google Colab. And protein can be modeled by AlphaFold. And you can create the complex of protein using ClusPro, protein-protein modeling. The binding site analysis by FTMap and docking using Autodock. You apply all these tools for the drug design. A quick review of submodule folder structure. You have 0 to 4 here. 0 module is basically how to execute PyMol and Autodock using the Jupyter Notebook. And majority of the modules contents are covered in 1 to 3. And within this submodule, we have submodule 1.1, 1.2, 1.3. We divided them to submodules for convenience. We have data files there. You can pull out the data from there to visualize. And some of the PML scripts are also there. And we have provided minimum number of PML scripts. The remaining, when you run these some of these scripts, it will be generated. What you need to know before you start this model, we assume that you have some basic chemistry and biochemistry knowledge, and you should be familiar with working in command mode on any computer. And before you start this model, you need to know how to create the virtual machine. Instructions are provided, and how to start PyMol and Autodock from Jupyter Notebook. Once again, instructions are provided. I will show you brief details of that. We need to know how to extract the protein sequences from the UniProt, how to download the PDB files from Protein Data Bank. Basically, just introduction about submodule 1. Here are the learning objectives and activities and visualization of proteins, as well as the option of using graphical user interface using PyMol, running scripts. And you will learn all the details of secondary structure, tertiary, quaternary, as well as primary structure in the submodule 1. If you go to the submodule 0, you will see how you can open PyMol as well as Autodock. I'll briefly show that, how to use that. So basically, command to start PyMol, you can go there and run the first cell. So once you do that, basically you can execute that. It will generate an IP address. You cut and paste the IP address in this cell. One thing you have to observe here is when you create such things, if rule already exists, then you can see here you have to change this. Accessories 1 is there. This happens when you are using this and go back to your home and try to use it. It's a different network. So as you change the network, you have to use this same thing here and here. So once you use that, the firewall will be created and you will get the command for the PyMol. So you can see the creating the firewall and a link for the PyMol. Once you execute this link, you will see that PyMol GUI is opening in another window. And in the PyMol, I have given the digital instructions how to use the PyMol. You can open different folders. In this case, I have shown S3 here. Probably in the actual one, you see bucket there. So different folders are placed inside for your convenience so that you can open the, you can see that you can go to submodule 1 and open the structures and heme group in the binding pocket, which is shown here. So that is helpful for visualization of protein structure. So once we have that, we'll look at how the Autodock can be opened in a GUI, graphic user interface model. So similar to the PyMol, commands to run Autodock. You can run these cells here and it will generate the IP address. You can see that you cut and paste the IP address there. So once you do that, you run the next cell. It will create the firewall. If not, once again, you have to go back and accessor 7 here, the same name here. You change the name every time you change the network. That'll work. Otherwise, it'll give a problem. So once you do that, finally run this block. And running this block will create a link. Hopefully, it'll take 30 to 40 seconds for the Autodock. You can see here, Autodock link is created. Once you open that, you will see the graphic user interface of Autodock. And with the help of instruction I have given, you can use the Autodock. So before going further, I want to share my other window to show you an example of a module here. So you can see that this is the module 1.1. We have basics of proteins, learning objectives, prerequisites, and introduction. We go through some basics of amino acids and basic knowledge. And each one, alpha helix, beta sheet, and so on. And then you go through the activity. You can see here each activity step-by-step instruction is given. Visualize the hydrogen bonding in the helix, and there are scripts there. You can go through that, and then you clean up. So hope that gives an idea about how to use this module. I would like to thank the funding from the Institution Development Award from the National Institute of Health and Dr. Lakshmi for support, my team. Thank you so much.